software, whether it's easy or hard to use, is often it's relative. When SeqMan Pro came out, 25 years ago, it was I think the easiest to use for looking at Sanger trace data. And really over the years, as GUIs have changed in software, and newer users started to find that same interface was getting more difficult. And we're finding that new users are reporting our classic SeqMan Pro to be quite difficult to learn to use. So we did redesign some of the interface, especially where you get started in SeqMan Ultra. So we'll focus there a bit today to show you kind of the entry points into the software, how we've kind of updated them to be more consistent with modern software. Regarding the hardware on the computers, we also, and this was more in last week's webinar, we find that people tend to use laptops more, smaller computers than they did just a couple of years ago. And so our software will assess the hardware on your computer and give you some feedback to let you know if you have adequate hardware in place. And we covered that more in the last week's webinar. So today I'd like to thank everybody first for joining us. And we'll dive right into the software. And what I'm going to attempt to do today is something I don't generally recommend, and that's have two versions of the software running at the same time, looking at the same files while the webinar is running. So it's a little risky doing that, but I think we'll be okay. But please bear with me if we run into any problems with programs fighting for the same views or anything. I am going to hide my webcam here just so we get a little more real estate here. And we'll come back at the end of the webinar. And I think everything is on the left side of the screen, so Sharon will let me know if you guys can't see what I'm talking about. So we'll try to keep everything here centered in your view. So a new point for the new users in particular, we've added something called the DNA Star Navigator. So when you first install our software, this navigator will appear, and we've changed the navigator. We had an older one back in 2019 that was replaced. And this new navigator is really a workflow navigator, and it will by default open like this. And so there will be molecular biology workflows, genomics workflows, and protein workflows. For this group though, we added a link, and that's organized by application. So once you learn which programs have the functionality that you're looking for, you really don't need a workflow navigator as much as just a nice convenient place to launch software. And so here is a list of our applications. We can launch SeqMan Ultra. So our new users coming into our software will usually go to the navigator. They may launch Ultra. And I'm going to hide this side panel here. So there we go. So SeqMan Ultra has a welcome screen. And this is something that will be consistent with all the DNA Star applications. So when you open it, it gives you options to open an existing project, start a new project, and so you can see they're broken down by molecular biology workflows, genomics and transcriptomics, or do some search and access training materials. By contrast, SeqMan Pro, and I'll launch it here. Keep your fingers crossed now. We're doing what I generally advise not to, but so SeqMan Pro opens really to some empty windows, right? And so we'll see we have an unassembled sequences window and an untitled window. So it's really not guiding us to do anything in particular. Of course, people who have used this forever know, well, to get started, I'm often gonna drag and drop my reads into this window, and that will let me get started. And the buttons at the top are really all the different parameters and settings that you need to set up and run assemblies of your data. This was designed for Sanger data. In about 2007, SeqMan Pro then became used for NGS 454 data, then Solexa, and then Illumina data. And so it adapted to accommodate the other sequencing technologies. What we found though is that because it was really dedicated and originally designed for Sanger, it works great for Sanger, not as good for some of the other sequencing technologies. So to give people better starting points, we guide them a little bit more. And I'm going to show you some of the guidance here in Ultra, and then also ways that we can shortcut some of the guidance too, because we don't want you to take too many unnecessary steps. because that makes people irritated if you have to click too much. So we're going to start by a new assembly, and we'll start with something very small, a Sanger ABI de novo assembly. And when I select this option, we're actually getting our SeqMan engine wizard is appearing here, and I'll make it a little smaller so we can see it. And the reason for that is SeqMan engine now has the most modern de novo assembly algorithms and the most modern templated assembly algorithms. And it has a wizard that guides us step by step through the assembly process. And one thing that we found is that new users in particular were struggling with some data sets. They would have failures because they didn't understand how to set up the parameters, or their computer didn't have the adequate hardware in place. And so we're going to provide some guidance here, and I'm going to show you how to shortcut it a little bit. So in a de novo Sanger assembly, generally, I just need to move this. I'm going to show you how to drag and drop. So there are some similarities here. Okay, I have to see my demo data folder. You guys can't see this on the far left, but okay. So I'm going to open up a folder that has some Sanger reads in it, and we maintain some of this functionality. For example, just make sure you can see that. I'm going to select these 14 ABI reads and drag them into the wizard, and we're actually ready to go. If I want to assemble now, I can click run, and I'm just going to do that. So I don't actually have to go through all the wizard. So the next page of the wizard gives me some options. So the wizard is more transparent. I now know about parameters that are sometimes very important. For example, removing small reads, doing quality trims, scanning for vectors or contaminants. And so these parameters now are very accessible. For some projects, they're critical. For a little de novo assembly like this, not as critical. So it allows us to run and bypass those pages. So it'll assemble very quickly. When I click finish, we'll get SeqMan Pro out of the way there. And now there's my assembly. It auto-named the project because I didn't go all the way through the wizard to set a name. So here's my assembly. And like classic SeqMan, I'll do the same thing just to kind of show you that there's a lot of similarity here. So we'll go to those same reads. And I'm showing you drag and drop because that's for people who have used this software for some time, that's the fastest way to get reads into the software. So you see with SeqMan Pro, I drag and drop. There's an assemble button here. The parameters are all behind these buttons. They're not really coalesced in one spot real clearly. There's some under options, there's some under project parameters. And when I've done webinars with customers in the past, a lot of times they didn't find this particular location, but this is really where the stringency is set for the assembly. Things like match percentage and match size are probably the two most important. And there's the old classic assembler and the newer SeqMan assembler called the Pro assembler. So there's actually two different algorithms with slightly different parameters. And we have the latest SeqMan engine de novo assembler, which is pretty similar, produces very similar results, but it's got quite a bit higher capacity to these assemblers. So I'm just showing you that the parameters in SeqMan Pro are in kind of different spots here. And you have to know where they are to go find them. But in this case, so there's some trimming end parameters. They're scattered a bit, but I can just click assemble too. I'm just going to let it assemble. And the similarities now in the UIs. So if I go back to that project report page, SeqMan Ultra gives me an assessment of the assembly, the config size, the number of configs, how many reads assembled. There's also a report here, right? This is the assembly report, and it tells me about how that assembly was, how it was run and the parameters. And likewise, SeqMan Pro, we have the config over here on the right, and then there's a report window. And SeqMan Pro's UI is different in that there's floating windows. They are not, they're not contained windows. They're all floating. But you can see that both programs will use this right side panel. In Ultra, we call it the Explorer, and it's really like SeqMan Pro. It's critical to use this panel to focus your selections. So if you want to perform particular actions, you need to select the config. In this case, there's only one config to actually run the appropriate report or to get the appropriate view up. So they're very similar, on purpose to design them. So the user experience at this point is quite similar. Now, to look at the assembly data, Ultra has some quick buttons here. So I can show an alignment, this first icon. So that's the alignment view that shows me the aligned reads. And then there's a feature table. And then there's a strategy view. And you'll notice that if I double or just click there, I get this view that appears. And now I can search this view, and I can also reveal traces. So we can look at the trace data. There's a coverage plot in SeqMan Ultra. So we found it cumbersome in SeqMan Pro. When I open a config here, and I'm going to double click, there's no short icon, and click the twisty triangle. And you can see that there is no coverage here. So I would have to get the comparable functionality, open two views, a strategy view and a coverage view. And you can see it starts, I'm a little compressed here in my space, but you have to have a couple windows open to kind of get the same information in Pro. And we found that that was awkward. So we just added a coverage plot here. And we have to navigate, I can use this zoom slider to zoom in and out on my view. And you can see when I zoom out far enough that I get arrows instead of reads, because we can't, and I can scroll and see there's my coverage plot where I can see the depth, at that particular area. Now, if I want to get back to where I can see my traces, there are some compact text and restore default zoom buttons that just bring me back to kind of a working zoom level. Now I have the resolution for the webinar. We set it so it's fairly low resolution here. So everything looks kind of big on a regular monitor that you're going to use. You can pack a lot more data into this view, but I can also use the compact text to get as much data into this view as is possible. Now another nice little feature here is that you'll notice that we have expand icons as well in SeqMan Pro, and you don't have to click on all of them, and we can have a context menu now just to expand all of them to see all the traces at one time. We had similar functionality in SeqMan Pro, but it wasn't very discoverable. And I did quite a few webinars where I would show people how to do that, and they said, well, I worked with this program for years and didn't know that if I hold, and I actually kind of forget, I think it's Control-Alt keyboard down. Yeah, Control-Alt, and I can get them to all expand at one time. And I think that's the only way you can do that in SeqMan Pro. And so otherwise you just manually kind of expand that way. I can look at, so we have a toolbar in SeqMan Pro where I can look at things like quality scores, right? We have something comparable in Ultra, where I can show scores. In Ultra, I can also, if I really want to make the peaks as big as possible, there's a vertical zoom scale, and also these data tracks. If you hover in the center of them, there's a grabber that allows you to really, make the peaks as large as you want. Okay, so and then you can kind of restore it back to the default. And so there's kind of our default view. Now, in a de novo assembly, we want to look for, oftentimes want to look for conflicts in the assembly, look for differences in the consensus areas where the consensus couldn't be called cleanly. And in SeqMan, there was a couple of ways you could do that. One way, which is a little bit in Classic, and this is one of the bigger differences between the two programs. In SeqMan Pro, we provided a variant report. And what this was really a difference of the aligned reads to the called consensus. So it wasn't quite accurate to call it a variant report. I mean, it is variations, but folks would use this to navigate to find problems in the consensus. But it's actually not, you can miss some of those areas using this table. So we actually removed the variant report because it was a little bit misleading in SeqMan Pro. And in SeqMan Pro, a better way to find these errors or problems in a consensus was really under Edit, Find Conflict, right? And it brought up this search window where you could set up some search parameters and then use the Find Next buttons. And if you notice down here in the left corner, there's these chevrons and it set the conflict. And now I can move through the assembly. I'm going to zoom out a little bit to find conflicts in the called consensus. And that's really the proper way to find all the conflicts in your consensus. And unfortunately, people would often go to the variant and actually miss quite a few of them because it's a slightly different calculation for finding these conflicts. So what we did in Ultra is we wanted to make that more discoverable. So we added a little search alignment icon here, and it pops up below the toolbar, and this allows us to search different ways as well. So I can go to conflict and use arrows now to move through my assembly to find those conflicted sites where the consensus couldn't be called. And of course, once we're here, we can do things like make edits. So we have, I'm just going to kind of just bring this back here. And I purposely chose a nice noisy, I didn't trim this data to leave a lot of, I left a lot of vector there just so we had a lot of areas to look at for the webinar. So this data actually isn't this terrible looking. So I can select a region, and by default, it'll ask me if I want to allow all editing. I'll say yes. So I can backspace and delete, and I can, insert bases. So same as with SeqMan Pro. I can, trim by dragging ends of sequences. So if we go, let's go find a ragged end here. So which is common with trace data is there's noise on the end, so I can do a little dragging here. And I will say, we're still working in some areas here in SeqMan Ultra to make improvements. One of them is just this performance right here of editing. There's a lot more going on with SeqMan Ultra, so every time you do an edit like this, it has to update a lot more than what SeqMan Pro did. So it's a little flashy. So that will improve in the next version, which should be later this spring. We'll have another major update to add some more improvements. And SeqMan Pro, it's been around for 25 years. We've been fine tuning this one for a long time. It's a great editor for Sanger data, and really we want to match what we could do in SeqMan Pro in terms of the responsiveness of the UI in Ultra, where Ultra can really shine. In addition to these small projects, is that it allows us to really scale up. I can also load a whole human genome in an Ultra and navigate it using these same navigation tools. And I can load much larger sequences and still get really good performance. So it's got to do a lot. It's got to, be editable for small projects, and it's got to act essentially as a genome browser for the large projects. And so we're asking it to do a lot, and we'll continue to be looking to improve it. Okay, so there's a de novo. Oh, one other thing, de novo, of course, you can export a consensus sequence out, so you can make your edits, export a consensus, just like you can with SeqMan Pro. Okay, so let's go back. I'm gonna reduce that navigator. One thing I really like about Ultra is I can open multiple projects. So here's this little de novo Sanger. I can start another project, and this time we're gonna do a ABI variant analysis. And I will point out that there's this other workflow, ABI reference guided. These two workflows are hard to differentiate from each other. And the reference guided is really a legacy workflow. We will, there is a way that we're gonna be able to replace this workflow. And what it does, it allows you to align to a reference and then make an editable output project. And whereas the variant analysis produces a BAM file that currently isn't editable, but we're gonna have a way to convert that, and that's part of this next update. So these two essentially will get merged into one workflow with the next major update. I'm gonna go to the variant analysis workflow now. Again, it's gonna launch SeqMan Engine Wizard. And you can see it's asking for the reference sequence. And here's where we really start to differentiate between SeqMan Pro, is that we have had these options for our NGS data for quite a few years, and that's things like downloading genomes let's see, from our cloud. So these are genome reference packages that have database information about SNPs. We can add from a cloud folder. So if you're logged in, you can have your data up on the cloud and load that data in. And also, we're not restricted in size. SeqMan Pro was RAM limited. You couldn't align, for example, to a human chromosome. It would run out of memory. And so this is using, the templated alignment is not memory bound at all, so we can use any kind of a template. So I'm going to go in here and pick, so I could pick chromosome. So this is a gene here, a GBK file, or I could align to this whole chromosome 22, just for time's sake. It takes about a minute to align to chromosome 22, and I don't really want to wait a minute here. If you have VCF files with lists of known variations, I can add them. So our Sanger customers now have additional capability that they didn't have before. VCF files are widely used to mark known variations, bed files to mark locations that have been targeted. So you can use the bed file then as a filter. If you've got an amplicon experiment, you may have noise on the outer edges of it where you had primers to generate your data, and you can use the bed file to easily mask out those areas that tend to be noisier. So we gain access there. Now I will say that the next page we add our data, and this is where there's also some more work that's ongoing right now actually, and that's to provide a different experience for multi-sample data. And I'll talk about that a bit when we look at the views, but we'll just keep this simple for the webinar. And I'm going to load in some Sanger data that's got lots of variations from some different samples. And we'll treat this as a sample. And so we can align this data to that gene reference or to the chromosome. You'll notice that there is no Run Now button yet. And that's something that we will have. Again, this data is still small. The gene isn't that big. We don't have to worry about hardware so much. If I had a chromosome though, where on a small laptop, that might be a problem if you don't have any space on a C drive, then you want to go through the wizard and have it make sure that your hardware has enough space to do a larger alignment. So you will see this Run Now button in the next major update. So it'll allow templated Sanger to be reduced down to 2 wizard pages would be the goal here. And I'll just to contrast that, we're going to go back into SeqMan Pro just so you can kind of see. So now I can't have these two, so I'm gonna have to close this project out. And just as a reminder how we did this here, and I'll go into, so here's my data, and I'm gonna drop this gene in. Now I could load chromosome 22, but it won't actually assemble chromosome 22. Now what you had to know with SeqMan Pro, again, the wizard isn't guiding me, so I have to pick a compatible sequence, and I have to use this button to mark the reference. And then the default setting for SeqMan Pro alignment is quite, for ABI files, is quite permissive. And so for most variant detection, you really wanted to go in and increase the stringency a bit. But we'll just kind of stick with default. So I'm just going to assemble this, and you can see we get our data, and there's our alignment. And this italicized sequence, that's our reference sequence. There's the features on it. And typically how you'd use SeqMan Pro then is you'd go to the variant report. Now it's a templated assembly. We can make SNP calls against the reference. We can launch a variant report, and these were interactive, right? I can select areas in the variant report. And you'll notice though that the variant report doesn't have a lot of information in it. And we're going to go back here to the column. So You know, I've got, just scroll here. You know, there is a codon change there, but it's a pretty basic kind of a variant report. And this is really our original variant report that we designed back in around the year 2000. So it was a kind of a home-brewed. We use nomenclature that isn't necessarily standard. So this is pretty outdated in terms of what it's displaying. There's a filter here, very kind of basic filtering. And so by moving to Ultra, we get to use the same variant report that we've had with our NGS data. And so now I'm going to click through the wizard, and you can see it's going to give me a little more feedback. What kind of variation detection am I doing? Is it a haploid or a diploid? If there's gender, I can add gender. If it's human, we can include the variant annotation database that brings in a ton of extra information for to human genome analysis. And then there's CNV analysis I can run, or structural variants. But here we're just going to run, and we'll just give it a name. And so here's the page that, if you're with us last week's webinar, that assesses what you're trying to do. It looks at your hardware and how much input data you have. And for Sanger data, it's almost always going to recommend run this locally. If it's de novo, you really can only run it. We'll only let it run locally. There's no need to run it on the cloud. If, for example, we have some Sanger projects where amplicons are targeted on various chromosomes on a eukaryote, then your reference sequence is going to could be in gigabases in size. And if it's a large reference sequence, you might not have enough disk space. It's going to estimate how much disk space you need and how much RAM for that particular assembly. And so if it is bigger, or you're aligning to a genome, then it might say, hey, run this on the cloud. It'll automatically upload, run the assembly, automatically download it, and it'll be ready for you. So it's a very nice way to improve the success rates as people scale up and do some larger things. So we ran the assembly. You see this log information that scrolls through, and the wizard now allows me to open the assembly. If it's multi-sample, if I'd set this up as multi-sample, and if you're a genomics customer, you'll know that you can go to another program to look and compare across all the different variations and get a nice summary report in a RayStar. Here, we're just going to open this up in SeqMan Ultra. And so you can see, I can see how many reads align, and we're just going to open up an alignment view and a Snip Table. And so at this point, the functionality is pretty similar to SeqMan Pro. And so we have a Snip Table, and now you see there's a little bit more information in this Snip Table. It does a genotype call. It detects if it's in a homopolymer. We get the DNA changes and amino acid changes now are in a standard convention. So it's a standard format for reporting variations. Now I like to have my SNP report down here. I don't know why that name came through so large. It's kind of funny looking, but so I can select, like I can in SeqMan, it's very interactive. I can select in the SNP table and it'll show that region in the alignment view. I can expand all to look at traces in those regions. That's a messy region. And overall, the SeqMan Ultra will be a more stringent, so it's better able to filter out noise at the end of sequences, and it does a little bit better gapping. So the accuracy, and in particular, the false positive detection rate is quite dramatically improved with SeqMan Ultra and using SeqMan Engine to do that assembly. So we can see I can collapse things down as we get crowded here, especially at this low resolution. You know, these buttons in the bottom right are really useful. So I can make this a single view and keep adjusting this view as needed, right? So I can also detach a view. So if I have two monitors, the way I would probably work with this is have my SNP table here floating and go set it over on my other monitor. And then I can really maximize my space, especially as you start to get bigger projects where these tables get quite large. You know, you can split the views and then reattach views. Okay, so that's some Sanger data analysis. I just want to show you one other project here, and I'm trying to. Oh, before I move on, there's also filtering dialog, and so the filtering dialog now is allows me to filter this SNP report. Again, it's unchanged essentially from the SeqMan Pro NGS version. So we have all this additional information to apply filters. So that user experience should be quite similar to what you have in SeqMan Pro. Now, of course, this is using the old. You can see how much there's just a lot less data that was extracted with the older version of the variant report. Okay, so I'm going to close this project, and here's where we get a little bit riskier. We're going to open something, a de novo bacterial genome assembly. It's going to take just a little bit, so I'm going to get this opening. This is an Illumina E. coli assembly, and it's got a bunch of contigs in it with some mate pair data, so we can make some scaffolds. So now a little more advanced use. So we looked at some smaller Sanger projects. When we scale up a little bit to genome size, I'll let that load, and I'm just going to keep this open here and open the next one. It takes just a little bit to open it. So now we have this de novo genome, and I edited it down a little bit just to make it open a little bit faster. So the contig N50, 139 kb. So these are some of the common metrics, largest contig size that you'd see in a de novo Illumina assembly. And these are just a bunch of contigs now that aren't in any particular order. And what we found is that in SeqMan Pro, the same kind of project really required expert hands to be able to work with the genome. And so we tried to take some of the tools and make them a little bit more discoverable. So there's some changes here in how the menus are laid out. You can see that in SeqMan Pro, I have these unscaffolded contigs, and under contig, I have contig editing type things, but then also under project, I have some scaffold creation. So I'm just going to create a scaffold here. Oh, excuse me, I want to order these first. So take that back. I'm going to go and order the contig. So it's going to read the pair information on the contigs and make scaffolds based off of that pair information. And so what we have now is we've taken that genome, excuse me, and we've made several contigs in the genome out of this. So scaffold 101, 111, 112. And the way that SeqMan Pro would work is we'd double click on a scaffold, and that's visualizing that scaffold. And so, excuse me. So navigating this was a little bit of a challenge. I had to zoom in, and you can see here's pair visualization, and trying to find the next contig in a large scaffold. I'm looking for what I'm interested in is the gaps. I've got gaps between these. And so you have to use the scroll bar, and it's sometimes hard to find where the contigs are. So navigating through this could be difficult. And then really what I want to do is close a genome. And I'm not going to go through all those steps, but it involved adding a sequence and kind of tricking it to add it into the project, and then reassembling the contig. And you had to use these locks here. So for example, if I wanted to work on contig 6 and 48, I would have to find the gap between them. And this probably doesn't look that great on your side, but so there's a gap. And I would change this to show me the mate pairs. I don't think it's actually showing them here. So I want to close that gap, and I would have to try to splice a sequence in. It's quite difficult to actually figure out how to do that in SeqMan Pro. So we made an improvement The locks here, folks didn't figure that out. And really the intention is don't allow sequence data to go into these other contigs. I want to lock them down and direct an assembly to a couple of contigs. And that's kind of an outdated concept, and new users we found really didn't figure that out at all, what that lock meant. So we changed the behavior. And when we look at SeqMan Ultra, we moved things and renamed them a little bit. So everything's under a config menu that's relating to the contigs. The project menu now is more limited. The view lets us open and close all the views. This organization of the menu is consistent now in our software. So if you go to Megaline Pro, it'll have a consistent organization. So we increase the consistency quite a bit there. And now I'm going to order these contigs. So it's, again, auto creating scaffolds based on pair information. And so now I have scaffolds, and I can double click there or use this icon that's a scaffold icon, right? And now I get a view that is a lot easier to navigate. I can zoom using a zoom slider, right? And I can find gaps, again, using my search. I can go from contig edge to contig edge, very easily go and investigate areas in this assembly. So if I want to go back to config 6 and 48 and look at that gap, and here's another the reason I want to get this project up is there's the other big difference between the two applications is that in Ultra, what controls how these views look is over here on these other two tabs. So Explorer tab, critical for focusing your selection. Style tab then controls, what does the alignment look like? What are the mismatches? What are the colors that are displayed? The strategy view, what kind of arrows are shown here in the strategy view? So in SeqMan Pro, that was all controlled under project parameters. And so you'd go into things like strategy view, pairs, it's kind of in this kind of a dialog. So we move that to be a little bit more accessible. So the style panel will control. So if I just want to look at the mate pairs that were used to make the scaffold, I can change it and now see there the mate pairs that made that scaffold. If I want to close a gap, if I have a PCR fragment that I think merges this gap, we've really streamlined that workflow. I can just simply select the gap and go to config, add sequences to close gap, and I get a little mini wizard, and the wizard will allow me to load a sequence in and assemble it, and it'll close the gap. So it's far, far easier to do genome type closure work in this new program. And I'll just cancel out of that. Then there's also a Trax panel, and the Trax panel has strategy view, alignment view, and reads, and you can see that I can control which tracks are here. So I can actually turn off data tracks. So if you want to customize your view a bit and not have as much showing, you can go into this panel and customize and make the view look like you want it to look. There's also, I should also point out, that we are still working on SeqMan Ultra. There's a couple things that aren't in place. I mentioned multiple sequence alignment views. So if you go back to kind of this project, we will have in the next version the ability to look at multiple samples all together in one view. And so if I have trace data from 15 samples, we can assemble them separately, align them together, and compare them more easily as separate samples, and then get reporting on those samples. So it'll be a really, really nice way to look at multi-sample data in this view, and it'll scale up. We'll have multi-sample exome analysis and genome analysis. So that's one area we're working. The other is features. So if I go back again to our little Sanger project, we will, right now in version 17.1, we don't have feature annotation. So if I select a region, I can't make a consensus feature here. So that's one of the bigger chunks of work that's yet to be done in Ultra, is adding features to that consensus or transferring features from the reference to the consensus, we're also looking to add more automated feature annotations. So we're going to try to address, not just have manual, but have some automated options as well. So with that, it's about 45 minutes in, and I think that we covered a lot of material there, and I hope that you have some questions, and I'll stick around and answer some questions. Thank you.
Okay, thanks for that, Matt. We have a few questions that are all about the same topic, which is there's a little bit of confusion about how the navigator shows the names of workflows, but people are not seeing SeqMan Ultra listed there. And also, once they get into SeqMan Ultra, and they're trying to start a new assembly, they're seeing a bunch of other workflow names. There's a little bit of just confusion about like, where do they see the application names versus where do they see workflow names? And are the workflows in the Navigator the same as the workflows in SeqMan Ultra?
Yeah, so let's go. So the Navigator is designed either by application or by workflow. And so if I go into molecular biology workflows, these include workflows that lead to assembly of data, or many of these actually go to Seq Builder Pro, like agarose gel simulation. And these icons, here's a couple Megaline Pro workflows. And then there's also just, these are actually launching points here. Sanger Project Analysis will just launch Ultra, but there's also assembly. If I click here, it'll bring me to the SeqMan Engine Wizard, right? And so it'll go to Variant Analysis Resequencing Engine Wizard. So the thought here is that new customers are generally the ones that are going to go through the Navigator by workflow. And so it will guide them to the appropriate starting point in the software. Current customers are much more likely just to launch SeqMan and go directly to the program. And because SeqMan Ultra is go back to this welcome page, because it's used for Sanger and genomics and transcriptomics, this page for new assembly has molecular biology and also genomics workflow. So if I want to do a genomics, Illumina de novo genome assembly and click, it will also go from here to the wizard, and it'll start me at de novo genome assembly. And then from here, I would go into the wizard and do Illumina de novo assembly. For these other workflows, we probably won't have the shortcut. For when we're doing larger things like this, you really need to have a wizard guidance to make sure that you have things, set up properly. So the workflows are not exactly the same. There's a little bit of transition yet, but the end goal is that they would be, the same workflows that you see right here versus what's on the Navigator.
And the other question is from a user who has been using SeqMan Ultra for a while, since it got launched a couple weeks ago, and they're trying to load SBD, Seq Builder profiles. I assume this is into NGen actually when they're trying to do an assembly, and they can do that, but they don't show up initially. They have to choose to display all files.
Yep, that is a known issue. It's been addressed in our development build, so our next update will have that. So yeah, it's not seeing that extension as a DNASTAR extension. So thanks for pointing that out, and we have it fixed. So the next update, you'll have that'll be working properly.
Great, and we have a final question that just came in, which is a user saying that after they are notified that their assembly has completed successfully, they click on the Open Assembly Results button, at the end of the Engine Wizard.
Yeah.
But the results are going into separate files. Each one is comparing the reference sequence to one sample. And she was expecting it to compare it to all of the samples. Is that an issue with the multiple project or multiple projects?
Good, yeah, good question. No, yeah, I understand. Yes, so right when you set up a multi-sample assembly in SeqMan Pro, what it will do is create a project, and each sample is in a separate config. And so to view each one, you would have a separate config. If I set up a multi-sample in SeqMan Ultra, it creates a separate .assembly output. And so they'll be open in separate tabs across the top as separate projects. That is what will change with the next update, is that those multi-sample aligned to a reference in the next update will align and open in one view so that we can easily cross-compare across. It's actually functionality we had in, similar to what we had in version 12.2 way back then, where we had kind of multi-sample viewing in the alignment view. So that will come back, but it's the next update that brings it back to where it really becomes a powerful analysis tool.
So that same user says, wait, sorry, didn't have my mic on. So that last user that asked that question says, wait, I have one more question. How will I be able to assemble those samples now?
So you can either choose to assemble it as a single sample. So for example, this assembly is 15 traces, and I treat it as a single sample in the setup in the project. So then all the trace files go together into one config. The other option is, and it depends if you're a genomics customer, you have DNASTAR, and DNASTAR can take all those different assemblies and merge it into one Snip project for analysis. So depending on your workflow, that in what software you own, that could be an option for you now. But that's something that we can help you with offline.